In this video, we will discuss about a personalized approach using microbiodata to treat uh, patients who are affected with cancer. So this is just a idea. I think this has not yet been reached to a point where it can be applied. So I'm not sure if it has been used anywhere, but this is the idea that is presented in the paper, where the patients who are affected with the cancer or who are diagnosed with the cancer, some of the studies can be done, that is their tumors can be analyzed or their blood can be analyzed, and to find out what are the mutations that are present and their microbial signatures, that is what are the bacteria that are overexpressed or downregulated in their tumors and how they are related to the pro-tumorigenic or anti-tumorigenic effect. And other things that can be done is to assess their immunity, that is we can take the tumors and blood and check what are the immune factors or immune related conditions that are present in them. For example, this how this PDL1 is one of the immune checkpoint uh, inhibitors, and it will suppress the immune cell response. And the CD8 plus cells, how this CD8 plus cells is positive cells are the one of the markers for the immune cells, how the immune cells, how many immune cells are infiltrated into the tumors, and whether the tumors has this PDL1 ligands expressed on their surface or not, and whether they are releasing IFN, and the IFN gamma is been present within this tumor blood of the patients or not. These other things also need to be taken into care, or these informations will also needs to be have in order to develop the personalized care. And the important thing considering this topic is about assessing gut microbiome and systematic inflammations. You need to check the patient's gut microbiome, how their microbiome is, and compared to the healthy patients, what are the differences it has. So it can be categorized into three categories. This unfavorable microbiome, it has that can be categorized as low diversity, and this resistance to these biological functions, resistance associated to biological functions, and unfavorable microbiome species which are either causing pro-tumorigenic effect or affecting the normal functions of the patients. And the treatable microbiomes, it is intermediate diversity. The diversities are intermediate, and some are favorable microbiomes and improve baseline biological function. These bacteria improve the biological functions, whereas in the case of unfavorable microbiome, the bacteria will resistance the biological functions. It will have a negative effect on the biological functions of the patients, and they can the targeted microbial interventions, it can, and these things can be improved. This treatable, this might changing the microbiome can be improved with the way the people are taking diet and probiotics. And the favorable microbiome is this high diversity, which has varying diversities, which can perform different functions, and they are abundance in terms of microbial species and highly therapeutic biological functions. So they can be used for more therapeutically target biological functions. So these things also needs to be understand, and what are the bacteria or what conditions the patients are in, and how, and these things can be changed by maintaining by changing or maintaining maintenance with diet and probiotics. And once they reach to this two stage on the right hand side, they can move further to have a better personalized care. And the last thing is this assessment of their lifestyle, how they are taking their diet, antibiotics, vitals, whether they have high fiber or low fiber, medical histories, as it is also essentials for the personalized cancer care. Because once they change their diet or they move back to their diet that does not have these compositions, then the personalized care will not have a good effect. So this is just a preliminary ideas which needs further modifications in order, to reach to a stage where it can be implemented on the patients. So this is just the idea that is published in the paper. If you need to understand more about this idea in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.